ahead and get started. Thank you.
Thank you, Marian, for that great introduction. I hope everyone can hear me well. Can you hear me? Great. And thank you for inviting me to Audubon, Minneapolis. I've been meaning to reach out and make this connection for a number of reasons. And one of the exciting things that I'd like to talk to you about is something I think that your membership would have an interest in and have helped the Bell Museum before. And so I hope this will be of interest to you all. And yeah, I'm really happy to be here today to talk to you about something I really love, which is museums. Let's see. So a little bit about myself. I grew up in New York City, and I didn't have too many opportunities to go out and explore nature or the world. I grew up in an immigrant household, and we traveled back to our to visit relatives in India, and then back to New York, and that was it. That's the extent of traveling I did. So all of the traveling and all of the exploration I got to do as a child and as an adult into graduate school was through museums in New York. As you know, New York has some of the best and largest museums in the world. And so I spent many of my weekends going to different types of museums and exploring the exhibits and really dreaming about the world around me. And when I went to college, I wasn't really sure what I wanted to do. My family very much wanted me to be a medical doctor, and I rebelled by wanting to do science, but not wanting to do medical science. And so I still spent most of my time as an undergrad not sure how I would pursue different aspects of science. And when I was about to graduate, I got a call from the curator of birds at the American Museum of Natural History offering me an internship. And when I went to visit and I started my internship, it was the first time I had walked behind the scenes in a museum, past the exhibits, up the elevator, and into where the collections were. And you know, the first couple of weeks, I didn't really understand what it was, but I was so excited to just be on the other side and be part of the coolness of the museum. And as I spent more time in my internship, I started to wonder more about what was in the cabinets. I started to open them. I started to look around, and it was just mind-blowing. I was so excited. And you can see in this picture, this is what a typical collection will look like. This is the Bell Museum collection, actually. And you open drawers and you just find these amazingly beautiful specimens prepared by people who clearly had a love of these animals. And every one of them tells a story. Every one of them has so much information. And so I was immediately excited by this and excited by the fact that I could start exploring the world through these specimens and exploring the nature and going places, thinking about where these specimens came from. What do they tell us about the places they're in and the species that they represent? And this was my aha moment, which is kind of funny because it's probably the reverse of most people's experiences, right? They see things in nature and want to explore it more. And for me, I saw things in museum cabinets and decided I wanted to spend more time out in the world and exploring biodiversity. So that's how I got into it. All of the research that I do is very much based on collection. So everything, all of the information that I get to study evolution, to study biogeography, to study genomics or morphological evolution, to describe new species, all of that has a strong background in collection. So all of the information, the data that I collect in my research has a specimen backing it in a museum, which is really powerful because not only do I get a lot of types of information, as I'll tell you about, but it will also allow me and other researchers to go back to that very same bird, very same specimen, and say, what other questions can we ask? Or, hey, did we get this right? Is there some other way to look at this? And so having a specimen in hand, it can be really important for that. But today I'm going to do something a little bit different. I'm not going to talk as much about my own research. I'll give you a few details about some of the things that I've done in my research, but mostly I want to use this time to tell you why natural history museums are important, right? I want to kind of tell you what gets me excited and what I think should be things that others who maybe don't want to spend all their time opening museum drawers, but are still interested in the information we can get from them. So I want to spend most of my time talking about that. And then I want to also talk about a new program that we're starting at the Bell, something that could be of interest to you and something that might be things that you can contribute to. And in my making of my slides and setting up this talk, I put together information from my research and a lot of information from other people's research. So this is a little bit different from how I usually give research talks. But what I wanted to say is I hope we can use these opportunities to ask questions. So if you have any questions, feel free to start putting them into the Q&A and in the chat. And I'll get to them at the end of the talk, or if you want, you're also welcome to raise your hand. I think that's a function you can have in this webinar. And I want to leave a lot of room for questions. So I probably, the things that I talk about will allow us to have ample time in the end. So feel free to ask me questions about any aspect of what I talk about here or aspects of natural history or ornithology, and I'll do my best to answer them as I can. So what are natural history museums, right? I think a lot of people think about museums and think about just the exhibits that they see, but the basis of museums really is the collections that they were designed to hold. So the Bell Museum, its history is that by state legislature, it was created to document biodiversity. And so the collections are the basis for the existence of the Bell. And over time, that has grown, to support the exhibits that most people are familiar with at the Bell Museum or all natural history museums. And the collections are also really key for supporting education. So the Bell Museum in particular is a natural history museum that is housed in the University of Minnesota. And we spend a lot of time thinking about education at the graduate level, training new researchers in biodiversity sciences. We also spend a lot of time educating undergrads using collections in our training of all sorts of scientists. And we start from K through 12 and educate all grade levels to think about how science can inspire them to do different things. Now, normally not normally, but typically people start off talking about giving talks about big subjects with a quote from an important person or a big name in the field. And I actually want to talk about another important person, a student that I got to interact with, and his quote for what natural history museums are, which I think really encompasses just everything that is exciting about natural history museums, right? So he says, natural history collections are not only important for preserving and documenting biodiversity, but they're also for research, education, and instilling an appreciation for the natural world in society. So they're doing so many different things in so many different ways. And I was so excited to see this quote from the student, because it instantly, I realized that people understand how exciting natural history museums are, even if they've spent just a few weeks with it, or a few months with it, or years. And so I think this is a nice way to summarize why natural history museums are exciting. And another thing I want to acknowledge early on is that the natural history museums are also have a history that we have to a past history that we have to acknowledge some of the past wrongs that we might have taken part in, as well as the acknowledgement that our existence is based on some aspect of things being taken away. So I'll start with this land acknowledgement that is from the Bell Museum, because this is land that used to be Minnesota Makoche. So the Bell Museum sits on the traditional and treaty land of the Dakota people, who, along with the Ojibwe people, are the indigenous peoples of the land now called Minnesota. And in recognition of this fact, and to honor the Dakota people for their care of and acknowledgement of this land, the Bell Museum waives general museum and planetary admission for Dakota and all indigenous peoples. And in fact, the Bell Museum goes further to support memberships, as well as waived or reduced fees for anyone who has financial need. So the Bell Museum is trying very hard to acknowledge past wrongs, as well as support people being able to access natural history, people who might not have all the resources of that. In my own work, I'm trying to think about how I can do this in terms of my part, the part that I play at the Bell Museum. And this is my draft of an acknowledgement of what past history as well as future needs are. And so in my mission to document, preserve, and interpret biodiversity, I acknowledge the past histories and cultures from where these specimens are originated, and acknowledging that in some cases these might be, sorry, my dog is trying to get and acknowledging that in some cases these might be harmful past histories that we have to make amends for. And I acknowledge the need to create an environment that welcomes and empowers people from historically underrepresented and marginalized communities to have equitable access to museum resources. And I think the most important thing is also to use these specimens to better understand the natural world. So these specimens have a history in terms of not just the biodiversity they represent, but the human history of when they came and when and how they got to be in the museum. And I think it's very important for us to make sure that we are doing the right thing by giving credit and by observing the respect that these specimens need. So the Bell Museum has 50,000 bird specimens, which is about what we would call a mid-size museum. To put that into context, there are about 3 billion bird specimens in all the museums worldwide. And about 45% of the specimens at the Bell Museum come from Minnesota. So that might surprise you a little bit, given that we think of the Bell Museum as Minnesota's state natural history museum. But the research that happens at the Bell and the specimens that are housed at the Bell are a product of the people who were the researchers who were part of the Bell Museum before. So the researchers have been inspired by other parts of the world. And, so we have a substantial collection from Mexico and from Russia and the Philippines and Canada, as well as other parts of the United States. And so what we're trying to do is being able to represent as much of global biodiversity as possible. And the way we approach that is by going to places that involve that are pertinent to our research. So the 3 billion bird specimens in all museums worldwide, keep that number in mind. I'm going to reference that again in a second. So how do we preserve birds? Well, we have different kinds of specimens that we use to document variation in different ways. This specimen, I don't know if you can see my my cursor here, but this is called a study skin or a round skin. This is usually stuffed with cotton. And what we're doing here is preserving the outside of a bird and preserving as much of the plumage, the bill, the feet, and the wings and legs, as the wings and tail as possible. And this can be used for a variety of purposes, but obviously what we're maximizing here is showing different aspects of the outward appearance of a bird. We also will often keep just the wings, and we do this sometimes because making round skins can be very difficult, but also when you see that in the round skins, the wings are folded together, and there's a lot of information we can tell from wings that are spread out in terms of how birds can fly around, in terms of their ability to maneuver in different situations. And so these sometimes having just the isolated wing can be really helpful. Also, birds can sometimes vary in terms of the number of feathers on on their wings, and how this can impact flight. We also keep skeletons, and this is a great way to look at variation in the bones. Egg specimens can be very important for a number of reasons. One is that egg shape, size, and color can be really informative to bird species. And eggshell thickness is also something that we're finding to be quite useful for looking at questions about the environment. So maybe you've heard of the case of DDT and bald eagles and other raptors. So one of the stories that I love telling the most is this of how we discovered the impacts of DDT, right? People started noticing that bald eagle populations were decreasing, and when scientists tried to figure out why, they noticed that the eggs were shattering when parent eagles were incubating them, and so they were able to figure out, using measurements from museum specimens and using measurements of birds impacted by DDT, that the impact of the pesticide was that it was thinning the shells. And if we didn't have museum specimens to go back to and compare it to, we wouldn't be able to have that conclusive evidence for the effect of DDT. And so sometimes we don't always know how the specimens will be used, but they can be used in so many different ways. And as a museum curator, one of my big goals is to think about what I'm doing with the specimen today versus what other people might be using it for. And what people might be using it for in the future. And that's the real thing, to think about what our future needs. We also keep nests. We take a piece of tissue for DNA analysis, and we use them for educational purposes by mounting them. And in some cases, we keep the entire specimen, the entire body, in alcohol and in formalin. And this is great for us to be able to see internal organs and musculature and other features like that. And so there lots of different ways that we like to, that we preserve, and each of them has an advantage for different things. And I'd be happy to talk more about what we can do with these later. So I said there were about 3 million specimens of birds in the world. In contrast, you might have heard this statistic that scientists have now estimated that there are 3 billion less birds in North America than they were about 50 years ago. So that's tremendous, right? 3 billion. So a whole order of magnitude more that have disappeared in the last 50 years. So the number of specimens in a museum are actually quite rare and quite important to tell us about what's going on with birds living in the wild today. So how can museums help us understand this question? How can museums help us address this really important conservation need? Well, as I told you before, each specimen is an archive and a record of a particular place and a particular time of a particular species, right? So each one tells a story. But the cool thing about museums is we don't have one of a species. We try to have a representation of as many as we can. We're often have records of species that are from different times and different places in their range. And this is an important aspect of comparing biodiversity. So if you think about how museums first started, it was in the Victorian era to think about how to kind of show the curiosity of all aspects of diversity. So they would have one representative of everything that they could get. In contrast, a research museum tries to have as many as possible. And we do this because there's a lot of value in comparisons. So, for example, one of the questions we often ask with museum specimens is, what are species, right? And this is a question that we can ask when we can compare across individuals, across their range, and notice if some of them might have slight variations in different ways. And we can use that to then think about, is there some sort of pattern in terms of the individuals, and maybe by geography, maybe by genetics, maybe by morphology, that shows us that there is differences to warrant being called a different species. Here's an example of this in a project that I got to work on, where there were species, and here's just one of each of them that were all called the same species. And when we went to museum collections, we noticed that there definitely are morphological differences. So one had a lot more white and reddish on its belly. One had very little white on its belly, and one had kind of a mid-range. So that could be geographic variation, and it could be just, variation that exists, and they're still, but they could still be interbreeding. Well, what we then did was use genetic data to test out whether these were different species or not. And we were able to show, and here's a phylogenetic tree where the 3 different types, the 3 different subspecies at the time turned out to be very different, to be very distinct genetically, and to have evolved from separate from each other for millions of years. We plotted the location of these individuals and noticed that they're geographically separated as well. And we used information from some of the living species to be able to show that they differed tremendously by song as well. And so, museum specimens can be really important for starting the initial hypothesis and using it to gain information about other aspects that will tell us whether these are different species. And often associated with museum specimens, we can figure out information about morphology, about genetics, about biogeography. There's usually behavior recorded with it. And in some cases, people are starting to also document the behavior of the living bird in terms of videos or song recordings, and associating that with museum specimens too. We can also use museum specimens to ask questions of how do birds vary, right? So we know that species can vary across geography, across time, and in different populations. And we can use museum specimens to measure different features to see how this change has happened over time, and how changes have happened between different species. And here's a great example of using museum specimens to collect this kind of data, but also to educate and train students to think about different ways of using museum specimens. Here are some of the students that I've helped to train, and we've been able to use museum specimens to measure different body features, to use imaging techniques to study bill shape, and we've done this in 2-dimensional and 3-dimensional ways. And an exciting new thing that we're working on is using CT scans to look at both the external and internal shape of various features. And I'll tell you a little bit more about that in a minute. And we can use this to look at whether some of the features are different, right? Sometimes when you're just looking at the plumage features, there might not be obvious differences, but once you start measuring things, you might find that things that, even though they might look quite similar in terms of plumage, are quite different in terms of their shape and size. And here's a study where we used morphometrics, these different measurements of the bill and the wing and the tail, and showed that these two subspecies that were these two populations that were thought to be the same species turned out to be quite distinct. And so just like in the other case, where we used first the morphology and then we went into the genetics and showed, again, with genetic information, that these are distinct genetically and have been evolving separately for millions of years. And so, you can use, there are different ways of measuring differences. And I think one of the coolest studies that I have seen use these techniques to really address issues of how species are changing over time, and this is over time in human lifetime, is the study conducted at the Field Museum. So this gentleman here in this picture is Dave Willard. And Dave is one of my heroes. He was the collections manager at the Field Museum when he started noticing birds colliding into windows. This was about 40 years ago. And he started picking them up and bringing them into the Field Museum and started measuring them and preparing them as specimens in the collections. And he did this for the spring and fall migration season for the last 40 plus years. And that collection is now more than, I want to say, 80,000 records of specimens of birds that have have traveled through the Chicago area. And we can use this to tell us how birds might be responding to climate change. We know climate change has been happening. And so what Dave and colleagues were able to do was to take the information from the last 40 years and show that actually just over 40 years, we can see significant differences in how birds are changing in body size. It turns out that birds are getting smaller. And their wing length is actually getting longer. So this will leads us to ask questions like, why? Why is that? Why are birds getting smaller, and why is their wing length increasing? So if they're overall getting smaller, we can perhaps make a hypothesis for why this might be due to climate change. Perhaps their breeding season is shorter, or perhaps there's more competition. And so these are the kinds of questions we can start to ask as we use this to study change over time. You guys might have seen in the news that the Chicago area had a really dramatic season this year. There was one day that resulted in almost 1,000 birds that collided into the convention center. So that's incredible. And that was unusual. That doesn't usually happen. And that one of the reasons it hadn't happened in a long time was because of the same kind of information. So these birds that Dave was picking up formed the basis of a study where they showed how many birds can be killed by buildings, and they were able to take it to the mayor at the time and propose that they change the policies for lighting buildings, especially during migration season. And they actually were able to pass some legislation to dim the lights. What happened in that day when 1,000 birds died was the convention center had some sort of event and forgot to turn off the lights. And it also turned out to be a day where the weather patterns were just so that birds were being kind of funneled into off of Lake Michigan and into this convention center. So it was an unfortunate set of circumstances, but one that I think we can learn from, and we have been learning from. And this is very similar to the Bird Safe project that was conducted in Minneapolis. Again, I will say that there are lots of ways to show the impact of birds, of humans on birds, and this is really an incredible resource for us to study not just human impacts, but also to study evolution and ecology. Another study that I'm really excited about, this was a study that I got to participate in, and it involves the student who had that wonderful quote in the beginning of my talk. This is a study that I got to conduct with a good friend and collaborator in India who had an idea for thinking about how barbets use their bills and to excavate, and he wanted to look at the shape of their bills, and he had some ideas for how different shapes in their bills might impact how they're excavating, whether they're scooping out the wood or whether they're pecking at it. And so but being in India, he did not have access to a lot of the specimens that he was interested in using. And this is where I could play a role. I was already doing a lot of CT scanning for my research. And so I was able to collaborate with him and help him scan these birds that are coming from, and a lot of these are specimens that are housed in American museums. And we the process is really cool. Here's a study where we took the museum specimens, we scanned them in CT scanning, and then we're able to use that to model what the bill could look like. And the bill, remember, is both the the bony part of the jaw, the extended jaw, as well as the keratin layer, which is the outer layer that we usually see in birds. And so that together forms the structure that really can that birds use to manipulate things. And using that model, my collaborators were able to build a kind of physical test of what happens if the bird is using the bill to peck for impact versus for torsion, which is to scoop. And so I hope I'm describing this as well as I can. But the idea and what's innovative about this is that, we're using we're using specimens that weren't collected for this purpose, but we're using them using modern tools, using CT scans, and using element analysis to really think about like all the different ways that we can understand birds. And so this was really kind of a high level way of thinking about museum specimens. And there are so many other studies that I can go into that do very similar things. And for because I don't have time, and I didn't put them in here, but I would be happy to tell you about some of these other exciting things that have really looked at coloration of birds, all the different things that we find exciting about bird diversity. They have an enormous array of colors, and it turns out colors can we've learned so much in terms of how these colors are produced, but also the physical properties of some of these different kinds of colors. So iridescence, so the shininess in a duck's wing can actually have incredible waterproofing qualities and things like that. It's just really remarkable how much we've learned in terms of the nitty-gritty of what makes birds so diverse. So I didn't go into this in too much detail because honestly, there's just so much we've learned in the last 20 years that I wouldn't do it justice, but I'd be happy to answer some questions or point you in the right direction if you're interested in that. But one of the specimens we have at the Bell that I really love showing off is this one in the center. These are evening grosbeaks, and you can see that on the left side is a male, and on the right side is a female, and in the middle is one that is half male and half female. This is a specimen we call a gynandromorph. And what's remarkable here is this one is shows very interesting bilateral symmetry, right? So it's right down in the middle, and bilateral symmetry is something that is quite important in most animals, and it's a feature that traces back to a very early step in development. So this probably happened when the embryo was 2 or 4 or 16 cells big. And so what's incredible is this documents the possibilities of what developmental changes can happen in a bird. This is a very rare specimen to have, and it tells us a lot about things that we can't normally observe without lots of manipulation. So again, specimens can be really cool for kind of preserving different aspects of the natural world. The other thing that I want to make sure people understand is when I'm talking about how much we can learn about birds, there's more there's more to an organism than just that organism. Each organism actually has a multitude of other things that are associated with it. And museum specimens can still preserve a lot of that information. There have been a number of studies that show that there are still lice attached to museum specimens. And so you can go in and actually comb through and find lice attached to the feathers of museum specimens. And these lice are often new species to science, and in many cases are very specific to that species. And so it tells us something about the coevolution of these organisms and how they have persisted over time in different environments. We can also tell information about diseases and the parasites and microbiomes within each specimen. And so we try to maximize the information content when we are preserving specimens in today's world, now that we know that there's so much more to an organism than just the one species that we see on the outside. And specimens also tell us where birds are found, which is really an important part of their natural history, right? It tells us the extent to where they live, where they breed, where they might migrate to. It tells us also where they have been found at different time periods for us to be able to then ask other questions. Why weren't they found in the past, or why are they no longer found in a certain area? And I want to give a shout out to a really great resource that the Bell Museum houses and has produced. It's called the Bell Museum Biodiversity, the Minnesota Biodiversity Atlas. And here you can actually find information about all of the specimens at the Bell. So you can actually go in and search by species or by a location and click through and browse around and find information about each of the specimens in terms of who collected it, when it was collected, and any other associated information. We are trying, as we move along, to also have pictures that document these specimens, and we'll have information about whether a specimen was used in a particular study soon, once we are able to compile all this information. But that would be ultimately a great way to think about how to take all of the information from these that we have about Minnesota biodiversity and be able to give it back to the community statewide. And there are also some really great educational programs designed around using the Minnesota Biodiversity Atlas. So if you're interested, I strongly recommend that you can go to this website and try to find that information. And if you don't find what you're looking for, please reach out to us and we'd be happy to help you. The other, one of the other things that I think is really important is that we are using museum specimens not just to study biodiversity, but we can also use it to study environments. So we can use them to study things like pollutants. I already talked about DDT, but we can also use it to study things that are perhaps not just pesticides or chemicals, but other types of pollutants. And here's another great study that I was adjacent to at the Field Museum. A lot of my stories go back to the Field Museum. It's an exciting time to be in a place where you can just, browse around, and just the research questions can sometimes just pop out. So here's one of those types of questions. Researchers noticed that there were some specimens that were just dirty. They were just, so here's two specimens of the same species, and here's what it looks like in 1996, and in 1906, it was really, really dirty. And so they wondered if this was because the air was dirtier at that time. This is around the time of the Industrial Revolution. And it turns out the dirt really is, it's soot, it's dust from coal burning factories. And they were able to use really high resolution imaging to be able to measure the amount of soot that is kind of stuck inside these feathers. And they were able to correlate it to when there was a high number of coal burning factories in the Midwest, and how the air started cleaning up after the passage of the Clean Air Act. And so here's an incredible record of the air quality of this region, just because there happened to be people studying the birds around there. This is, I think, one of the coolest uses of museum specimens that is not about documenting biodiversity. So I really love telling this story. So what can birds tell us about other species? So as I said, it's not just information about that particular species or specimen, but each of them also carries a record of what they ate. And that is a record that we can get from their stomach and gut contents, obviously, but we can also get it from their feathers. And so using samples of feathers from historical times, so going back about 100 years, scientists have been able to show that in some cases, diets have changed over time. So for example, insectivores' diets have changed tremendously, and it seems to correlate well with the decrease in insect abundance that we see today. So sometimes museum specimens of insects themselves might be poor in representing this diversity, but we can use analogs such as birds to be able to get to some of these questions. And then finally, I want to say what we can use museums, we can learn from specimens, but we can also use specimens to learn, right? And I should apologize if anyone's sensitive to these pictures, but it might be a gory thing to think about dissecting a dead animal, but you learn so much, right? You learn so, and this is an internship program that we started at the Bell Museum, where we were training high school students to think about the opportunities they can have in a museum. And we taught them how to take data from the museums. We taught them how to dissect them. And we taught them how to talk to the public about this this process. So talk about why museums are important and how we can do that. And they had so much fun. You can see from these pictures, they're engrossed. We learned a lot about different species. We also learned a lot about each other and what we like. And it was a really fun way to engage students. And I just wanted to point out that I was going through some of these pictures and found this great picture of Bob Jansen, who came to visit us during this this event. And I wanted to acknowledge just how important it is to have people like him and others who have who've just dedicated so much of their time to understanding Minnesota biodiversity. And so I do want to acknowledge Bob, who passed away last week. And then continuing on this, how do we use specimens to learn? We can also, there are many ways to learn from specimens, whether you're actually holding it or not. One of the amazing things with in today's world with imaging and computing tools, we can take pictures. We can take pictures in different ways, and we can use this to distribute information to people who don't have access to a natural history museum in their vicinity. And this is an important way of kind of giving back to some of the communities where some of these specimens have come from. Sharing data across across the globe is an important way that museums can really contribute to increasing access and increasing inclusion in the field of biodiversity sciences. And there's tons of of databases now that exist that have information like this, that compile information from different museums and are distributing data that in an open way, in an open access way. And so if there are people who are interested in learning about specimens, there's lots of ways to do that, and there's a lot of information that's available currently. And so if anyone's interested, I'd be happy to talk more or point you to the right resources to find this kind of information. And these are great learning tools. These are great research tools. And so I really want to say there's been a concerted effort by many people to make these kinds of resources happen. And I think it's going to be really tremendous going forward in terms of allowing people access to data that they can use different in ways that are more innovative and than people who contributed to it could even dream about. But we also want to build on these collections and be able to acquire them for continuing to build on collections and continuing to learn from specimens. And there are many ways to do this. We can do more field surveys or go back to places we haven't been to, go to new places to discover diversity. But we can also think about the ways that we can collect them through some of the anthropogenic sources that I mentioned before. So this is a picture from BirdSafe, which was a program that Audubon Minneapolis actually had. And this was before my time at the Bell. And I would really like to kind of wrap up my talk by talking about a program similar to BirdSafe, where we're using the idea of salvaging wildlife, things that might have died because of some sort of human impact, whether it was colliding into buildings or cars or some other way that we find dead animals. We are trying to create a resource by taking all these tragic deaths and turning them into museum records. So this is a project that was funded by by LCCMR, by the Environment and Natural Resources Trust Fund of Minnesota. And we are the project has a couple of goals. We're trying to build a network across Minnesota that will that includes institutions as well as individuals who might come across some of these dead animals and are willing to bring them to the Bell Museum. And then we'll use these specimens to collect standardized data about the specimens, about diseases and pesticides, and use that to research questions about their evolution, their ecology, and about the environment, and about environmental health. So we are trying to build a way to take each of these specimens and make them useful in multiple different ways. And like I said before, we will learn a lot about this. So we will be able to use this to provide information back to the citizens of Minnesota and inform them about trends that we see and educate them about the value of studying wildlife in different ways. And this is something that I think all of you could contribute to if you wanted to. If you find a dead bird, it would be really great if you were able to pick it up and bring it to the Bell Museum, and by including some of the data that come including some of the data such as where it was found, when it was found, and who found it. And if you can keep it in a freezer until you can bring it to the Bell Museum, we'd be happy to take such specimens. And again, I think this is one of those ways that can help us a lot in terms of some of these, taking some of these tragic events and turning them into data points, and turning them into ways that we can better understand biodiversity and better understand the natural world, and better help with conservation needs, and figure out how we can coexist with nature in a more in a way that is friendly for everyone. So I think I'll stop there, and might have talked a little slowly, so I might not have left as much time for questions as I thought. But if anyone has any questions, I'd be happy to take them.
Thank you so much. So if you have any questions, just type them into either the chat, the Q&A box, or you can use the raise hand button, like Dr. Reddy mentioned earlier. I can kick it off with a question just regarding the wildlife project that you mentioned about donating dead animals that people might find. For birds, do you want only like native Minnesota birds, or like if someone were to find a pigeon or a starling or something like that, are those also something you're looking to collect?
Yeah, that's a great question. Honestly, we often get people thinking that we only want the rare things, and that might have been true in the past. But because we're now trying to document as much as possible in everything, we would love some of the things that people see commonly as well, because some of the common birds are going to have as many stories to tell about the environment. And so we anything that looks relatively fresh, we don't want things that are super decaying, because that's going to be harder for us to be able to collect a lot of different kinds of samples from. But things that look fairly fresh, things that probably died in the last couple of days that were already predated on, would be excellent.
I have a couple other questions while I'm waiting for people to type theirs in. For the slide where you talked about like learning about bird diets and insectivores and environmental insights that you might get, I was curious about when you said that there's like fewer available insects for certain bird species, and just like when you said that like the air has gotten so much cleaner through regulations since 1906, I kind of am thinking of like the 1950s, like the Green Revolution, when there were like pesticide campaigns everywhere. And I feel this anecdotally, there's like more like no mow May and native plants. And so I was just wondering if you could talk a little bit about why we might be seeing fewer insects even after all of these new regulations and like a growing social awareness.
Yeah. So I am not an insect expert, so I might not be able to answer that very knowledgeably, but I will say that some of these legislature and some of the legislation to not use as many pesticides is fairly new. So we're finding that kind of similar to what happened with DDT, we solved that problem by outlawing DDT and things that were having impacts on bigger animals. But we found other substitutes, other pesticides that were still killing off the small things like insects. And I think the impacts of that are really hard to recover from within a few years. So I think it's going to take a little longer. And as you might not have noticed, because it was kind of small on the slide, but some of those were trends that were happening over the last century. So this has been happening for a long time, right? And so sometimes it's hard to, the recovery period can take a lot longer. And we also don't know how many insects might have gone extinct in that time as well, whether locally extinct or globally extinct. And so I would imagine that a lot of that is probably because we're just shifting the kinds of chemicals we're using. The other thing is that, I mean, the vast majority of land still is destined for agriculture, right? We haven't restored a lot of the land that these insects and the things that depend on them, like birds and other animals, really need. And so, you know, there's some really horrible statistic about how little prairie land there is left in Minnesota, right? And so if you think about how much used to exist in the times in like about 150 years ago versus now, it's pretty incredible. So,
And then do you have any leading hypothesis for the wing length trend towards longer wings and smaller body size, particularly with the longer wings?
yeah, so I hope I get this right, because this is not a study that I was involved in, but I think it's really, really cool and important. So the idea is that the body size is getting smaller, and to compensate for the um, the what it takes to migrate, birds have had to like develop longer wings, because it is quite taxing, and quite a lot of it requires a lot for birds to migrate. And so if they're going to be smaller overall, they have to like somehow compensate for still being able to make that same journey. And so they think that that might be why it's kind of the trade-off between becoming smaller but also needing just as much to be able to make that journey. So they're still working on figuring out all of that, and it's hard to test that, but it is one of the hypotheses for why that might be.
So interesting. We do have a question from Wilmer, who asks if you have had groups from the public visit the bird specimen collection, and if it's possible for the public to see it as part of a public program.
Yeah, so that's a great idea that other people have asked, and we are considering that actually. So we would like to be able to do that. Maybe start with small groups. But right now, the main challenge at the Bell Museum is capacity, right? We don't have enough people to be able to organize this as a regular thing. But we do love doing programs where we show off some of our specimens, and we would like to eventually do some public tours. So I do appreciate that question. If you're interested, you know, we do some programs at the Bell where in the summer we do the dissections, and in the so there's a program we call Expeditions to the Bell, where we do dissections in the summer outside. And then in just last weekend, actually, we did a program called Collectors Day, where we brought out a bunch of specimens from our collection. But I would love to be able to think about having public tours. And something else I forgot to mention is if people are interested in volunteering, the Bell Museum is always looking for volunteers as well. So if that could be another way for people to contribute to collections and research.
Thank you. And I have another question about just your background as a women in STEM and first generation American, and you're a PI, you have a lab and you're faculty at a university. And I was wondering if you could just talk about like the impact of that on increasing the diversity in the like science to academia pipeline, and maybe just speak to like you talked a little bit about the amazingness of biodiversity among birds and why diversity in science could be important for novel research or new discoveries.
Yeah, that's a great question, right? And I think diversity in science, and diversity in anything, and we're finding this over and over again, is people who come from different backgrounds ask questions in different ways. And they're able to see things from a different perspective that just can connect. And often it's very it illuminates and broadens perspective more than anything, right? So I trying to find an example that's not just personal, but one that is somewhat personal to me is, I didn't come to study birds as a bird watcher. I started watching birds after. And so I didn't have a lot of kind of preconceptions of what I thought birds could were I didn't have a lot of preconceptions of so I'm an evolutionary biologist, and I didn't have a lot of preconceptions of what birds are related to. And so when I started doing my research, I would get results that were somewhat astonishing, and people who were birders had really strong opinions on it must be this and it must be that, and that doesn't make sense. Your analysis doesn't make sense. And so it took a long time to convince people that, I didn't bias the analyses. This is what comes out when you put in all of the data. And I was open to like thinking about different relationships in a way that people who kind of had these ideas based on what their hypotheses were for a long time, really took a lot longer for them to accept some of these findings. And these findings have later, these findings were shown over and over again to other people found the same things. And so, just not being able to like kind of expect a certain result because that's just what you learned before, or that's just what you had assumed before, is really important. And there are many ways to approach the issue of diversity and inclusion. I came into this field because I was looking for a subject that I could contribute to. And, there was something that just really sparked in me when I went into a museum. But and I also came from growing up in New York City, where museums were just this place of wonder and excitement. But I can see how sometimes museums can also be perceived as really harmful places for people from certain backgrounds, for people whose ancestral place was stolen from them, or people whose resources were stolen from them. And I have friends who talk about how some of those stories can be really inspiring, but also really hurtful. And so it's really hard for people to kind of like process all of that when it comes to museums. But if we can think about, in moving forward in a way that will be respectful of what that past history is, as well as thinking about like being respectful that this bird gave its life for us to be able to learn something more. So I try to think about it from multiple angles like that, but it's definitely a lot more complicated in terms of really thinking about the value of museums. Yeah.
Yeah. I have a couple questions about extinction and endangered populations. You mentioned with the bluebirds, I believe, that you were able to was it bluebirds? With the different species and like speciation through like genetic differences. And I was wondering with like extinct birds from a collection that are like of an extinct species or an extremely threatened population, if you can see that like small, like shrinking gene pool happening as like their numbers decrease, like can you see population stress in individuals?
Yeah, we can. So we can actually find that signature even after the population starts going back up in size. So we call that a population bottleneck. And genetics is actually a great way to really approach that question. So, obviously when populations shrink, you can count the number of individuals in a population, but like the impact of a bottleneck can be something that happened even like 10,000 years ago or 1,000 years ago, and we can pick up the signature of that in their genetic variation. And so there have been some cases that people have been really surprised by the results because they're for example, cheetahs are not they're not small in terms of population size today, but we can tell from their genetic history that they had a population bottleneck in the past that has really reduced the amount of variation that's within cheetahs. And so the same case, we can say, trying to think of a bird example right now, but one of the projects I'm working on now is the Great Lakes piping plover. And this is a species that is highly threatened, highly endangered, and went through a very severe bottleneck of, it was down to less than 20 pairs in the 1980s. And so we now have about 70 to 75 pairs, which is still not a tremendously, it's not large enough for us to feel comfortable that this bird can survive the next couple of decades. And so there's still a lot of management that goes into caring for and protecting this bird and protecting it from some of the threats that it faces in its natural range. But we are now looking at their genetic variation to see how much the bottleneck affected what their capacity to adapt to new situations can be. So genetics can be really important for looking at different aspects of that. So I appreciate that question.
Oh, and I have another one that might be a little off the wall, but as a scientist, have you seen enough compelling evidence to believe that the ivory-billed woodpecker should not yet be declared extinct? And then as a follow-up, like, I know that the declaration of extinction has some like funding implications. Like, do you think there's an advantage to keeping that question open?
I got a t-shirt from some students last year that said, I want to believe.
Yeah.
It's really cute. I think I appreciate in the case of the ivory bill that it's gotten people talking, and it's a great public education example, right? It's got people thinking about it. People know about the ivory-billed woodpecker, whether they know about birds or not. They know about extinction. And so I think that because it's gotten people excited to talk about it and to understand the issues, that it's been a really important topic. Whether there's enough evidence to say that they have persisted, I'm fairly skeptical. I'd like to, I would be very happy to be proven wrong. And I would really like to see some evidence that's more
photos?
Yeah, that's more than fuzzy photos and videos. That being said, like one thing that makes me really sad is if they are still persisting, it's kind of like, despite everything humans have done, they have managed to survive, which is not quite the conservation success story we want to tell, right? And so I think we should be doing what we can to protect that area, protect the habitat, and kind of maintain habitats for different kinds of species. One thing that we can learn from the ivory-billed is our policy of taking out all dead trees shouldn't be the standard way of doing it. There are lots of biodiversity that depend on these kinds of these things that as humans we see as, oh, that's a problem, we need to cut it down. And so, we have to start thinking about ways that we can live more sustainably and ways that are more friendly to other organisms, not just what our needs are. And so I think we can learn a lot from these kinds of examples. And again, I'd love to be proven wrong, but I'm a little skeptical.
Skeptical too. I think part of it is like even the most rational scientists can be like irrationally in denial about what we've done to a bird species, and it's hard to probably accept that they're all gone, even though that seems like what the data are telling us. I have another question that might be a little silly if you have time.
Sure.
And I am monitoring the chat, but I have a lot of questions even if no one else does. So with the genetic bottlenecking, and when you were mentioning different species that have survived that genetic bottleneck and been able to like revive their populations, I was reading about this project in, I believe, the Black Hills or South Dakota, somewhere in the Great Plains, where they cloned from like a collection like yours, but of black-footed mink, not to like reanimate an extinct species, but to like create a second like gene pool to like increase the like robustness of that breeding population. And I was wondering if that's like cost effective or like a good return on investment for like really vulnerable populations like the California condor or other really struggling species.
Yeah, that's actually a great question. And I do talk about this example in my class. I teach a class on conservation biology. And what you're talking about is, we call it genetic rescue. And this was in black-footed ferrets that are highly, highly endangered. And they've had a captive breeding population for, I want to say several decades now. And it hasn't been successful enough for them to stop intervening. And so they're thinking about other ways of helping with that. With the California condors, they were really lucky that they had to do something similar in terms of adding some variation to the gene pool, as well as and so we would do a genetic rescue for those reasons, to add some variation. And the numbers for California condors had gone down to such low numbers that they were just worried that they wouldn't even have enough individuals to breed with. And so they were lucky that there was a closely related species, the Andean condors. So they were able to take some individuals of Andean condors, their closest relatives, and mix them with the California condor. So I believe we think that's been very beneficial for that particular case. And they've now reintroduced California condors to places like the Grand Canyon and a whole bunch of other places. And so they're actually doing pretty well in the wild now. With the black-footed ferrets, I'm not sure. I think part of it is that there aren't any close relatives in the same way as the Andean condors. But and I don't know in terms of success. I'm not. I don't have the most up-to-date information on that. And part of the other reason why we do this is because, when populations are really small in number, we worry about several things. One is that they can get wiped up really easily, right? There can be a disease that goes through that they're all susceptible to, because they're all genetically related, or they're all in like a very small area, and so they can be able to transmit that disease really easily. So that's one big worry with endangered species. And then the other is that, you know, we want them to be able to persist and in the future, and the environments are constantly changing. And so they need to have enough capacity to adapt to these changing environments. And so if they don't have a lot of variation, they can't adapt to things like climate change or other aspects that might perturb the population. And so whether it's cost effective is really a function of how much resources we're already putting into trying to help these populations, right? And so it's a hard call between extinction and doing everything we can.
Yeah, I guess I just meant like, I didn't mean like, is it too expensive to save them? I meant like, in a limited pool of resources, if we're gonna try to save as many animals or species as we can, is this like a good way to go about it?
Yeah, no, I think it's a good ethical question that a lot of people ask, actually. Yeah.
Okay. Is there anything that you've learned from your collections that really surprised you, that were unexpected? Or
Yeah, I actually I'm I'm a complete museum nerd. I love like going and opening cabinets and finding new things. And sometimes I'm not doing it just to explore. I'm actually doing it because somebody asked for something, or we were wondering if there was any specimens of a particular species. And so every time I open a cabinet, I learn something, and I'm amazed by it. I love looking at the actual specimen with the of the bird, but I also really like looking at the tags. And sometimes you can get a lot of information that from the tags of people who've actually noted what the bird was like before being stuffed. And there's also a lot of information sometimes about who collected it, or how it was found, and all that. So I really find that stuff fascinating. So I can't think of a particular example right now, because I get excited by all of it. I get excited by the old ones and the new ones. People, when we do public programs, are always amazed by how good some of the specimens that are more than 100 years old look. I find that amazing too, because feathers preserve really well, and it's incredible how much we can get from specimens that are 100 years old or 150 years old.
that is amazing. The 1906 specimen, I couldn't believe that just from the photo.
Yeah.
And that even the soot is still intact. Like, you would have
Yeah.
think it was like rolling around in the drawer and would have gotten like a little less dusty, but that's so remarkable.
Yeah, sometimes I think it's good that we don't over clean things, so we can
Yeah,
actually figure out some of those questions. Yeah, I'm blown away by some of that too, like the fact that people can go through and find lice on birds that died 100 years ago. And then the lice are still preserved there. And still like attached onto the feather barbs, and they can pull them off and study all that. I think it's fascinating.
that is fascinating. And I think we're down to our last question, which is no question, just want to say thank you. And so I also want to thank you so much for teaching us about the Bell Museum and the amazing things we can learn from these collections. And just remind everyone that this will be posted on our YouTube channel. So thank you so much, Dr. Reddy, and I really appreciate you coming here to teach us tonight.
You're welcome. Thank you so much for having me.
All right, take care, everyone.
Bye-bye.
Bye.